Thank you guys for coming over. People who came over, and for those who are online, thank you for joining us. Sorry, we're getting started a little bit late. We had some technical issues and clinical issues and all sorts of issues. But we'll get started now on what is an annual event. We have annual R25, now UE5 grand rounds. We've been doing it now for 15 years approximately, that we've been doing this as a chance to highlight the work that the residents and fellows who have been training under the R25 UE5 training grant can show you what they've been working on. And just so that you know, this change, just so you get it into your heads, we are now calling this a UE5. The NIH, in their wisdom, about a year or two ago, changed it from an R25 to a UE5 for reasons that I have no idea about, but that's what we now call it, just so we keep it in mind. So we have three speakers today. Simon Gritsch, Peter Hadar, and first Yedda Li will be talking to us. Yedda is a fellow in the neurogenetics and gene therapy program here at Mass General. She's been working under Florian Eichler and Benjamin Kleinsteifer on genome editing strategies in neurodegenerative disorders. She's been on the UE5 for a couple of years now, and also has a Spark Award from the MGB Gene and Cell Therapy Institute for her work on genome editing. As an adult neurogeneticist, Dr. Li will serve the rapidly growing number of adult patients with neurometabolic disorders, and she hopes, and I expect will, run a lab to design effective gene-based and mechanism-targeted therapeutic strategies addressing the overwhelming and urgent needs in this vulnerable patient population. So without further ado, let's have her come up and, let's see, I'm doing this correctly. Talking about precision genome editing for microgliopathies. Thank you.
Thank you. All right, hello everyone, and thank you for being here. So, disclosures, consultant for visual neuroscience. So let's talk about microgliopathies. They're grouped into two categories. They're primary microgliopathies and secondary microgliopathies. The primary ones are inherited disorders, monogenic, affecting the microglia, specifically as the primary mechanism of pathogenesis. Secondary microgliopathies are other neurometabolic disorders that affect the microglia and change how they activate and how they regulate the neuroimmune system in the brain. I put this slide here mostly to say that microgliopathies have a lot of different phenotypes, but as a CNS disorder, or as a group of CNS disorders, they are amenable to genome editing, targeting specifically the hematopoietic system. So microglia come from two, I guess, different origins. One, you have native microglia in the brain, but you also get macrophage-derived microglia-like cells, MDMI cells, that come from the hematopoietic system and the bone marrow. And on the right here, let me turn on the pointer. There we go. Can you see that? Sort of. Yep, there we go. Okay. So on the right here, you can see that as we age, when we're fairly young and the blood-brain barrier is totally intact, you have fewer MDMI, and as we age, or as people get neurodegenerative disorders, the blood-brain barrier really starts breaking down, and you get more and more infiltration of these MDMIs. And so this creates a situation in which we can actually target the hematopoietic system and use the hematopoietic system to treat CNS disorders. And this has already been shown. So all of these disorders with check marks next to it are known to be able to be effectively treated by hematopoietic stem cell transplantation. And not only is it allogeneic transplantation, but diseases such as MLD and X-linked adrenoleukodystrophy have already had lentivirus-mediated gene therapy targeting the hematopoietic stem cells and facilitated autologous therapy. So let's talk a little bit about genome editing technologies. I'm going to focus mostly on base editing today because that's the data that I'll be showing you. But the two primary and the two initially developed methods are base editing and prime editing. With base editing here, there's two important parts of the base editor. you have a deaminase part that deaminates cytosines or adenines, cytosine base editor or adenine base editor. And you have a Cas9 nickase derived from S. pyogenes that basically makes a nick in the DNA in a very specific location. And that facilitates either C to T base editing for the CVEs or A to G base editing for the ABEs. And how this entire complex knows where in the entire genome to go is with this PAM sequence at the end and a guide RNA. So the guide RNA is complementary to the place in the genome you want to go, and you must have a PAM sequence to target to that area of the genome. So let me talk a little bit about the PAM sequence. So the wild type SP-Cas9, the PAM sequence that must be present is NGG. So as you can imagine, that really limits the number of places in the genome that you could target because it has to have an NGG PAM. So Dr. Klein-Stever's lab basically are making, or have already made, I should say, this is published quite a while ago, but have made more freely targeting PAMs. And SPG targets NGN, so that frees up one more base. And finally, SPRY, SPRY, targets NRNs more than NYNs, but basically you can now freely target throughout the genome. And this is really important because as you can target more places, you can correct more mutations. Okay, so let's talk about genome editing specifically for the microgliopathies. Again, I'm going to be mostly focusing on base editing here. So there's a variety of different ways in which we can deliver the base editing technologies. Most of the initial studies used electroporation. Now you can also use mechanoporation for cells that are more fragile. You can use viral mediated delivery, AAV usually mediated delivery, although the issue there is that the AAV payload is very small, limited to a very small 4.7 kb, and that's not usually enough for the entire base editor. You can also use lipid nanoparticles, and there's tons of studies out there now looking at lipid nanoparticle delivery. I'm mostly going to be focusing on electroporation today. And then the target cell type. So for CNS disorders, you can deliver directly into the CNS, but right now technology is still kind of limited in how efficient that is. So again, I'm going to be talking about delivery into the hematopoietic stem cells. Okay, so the platform that we've made here is that you do an initial screening using human cells. So you use human reporter cell lines, HEC293T reporter cell lines, and you can also use patient primary cells. So the HEC293T reporter cell lines basically encode various mutations in a gene or in several genes that you want to target. So it contains the mutation and a couple hundred base pairs on each side to look at bystander editing, which I'll get to in just a second. Can also then test in patient-derived cells. The readout here, of course, you want to look at on-target editing in the green here. So for example, if you want to edit that particular C, but the guide RNA that you use kind of extends about, it's like a 25 mer that extends out here. And you could, depending on the editing window, get edits in areas that you don't want called bystander editing, and that's in the red. And again, after that, you can also get something called off-target editing, which is just editing somewhere else in the genome that is not related to this site that we obviously don't want because we don't want to introduce random edits into the genome. So after you look at HEC293 T cells, after you look at patient cells, now it's time to look at patient CD34 positive hematopoietic stem cells because this is the eventual cell target. And what we want to do is first do animal studies to show that one, edited cells do engraft, two, that edited cells do reconstitute the human immune system, and three, it does create a functional, the edit does create a functional protein. So the disease I'm going to be talking about today specifically is ALSP. And I want to talk a little bit about why ALSP is a good disease to model this in. As a first choice. So one, it's one of the most common leukoencephalopathies, leukodystrophies affecting adults. it's massively underdiagnosed and misdiagnosed. So I suspect the reported prevalence is actually much lower than what the actual prevalence is. It is a primary microgliopathy, so you're targeting the microglia. There is published evidence for HSCT. So CSF1R is the gene that is mutated in ALSP. It is an autosomal dominant disorder. And overexpression of CSF1R has already been associated with multiple cancers. So if you use something like gene therapy, like lentivirus-mediated gene therapy or AAV-mediated gene therapy, usually you get overexpression or superphysiologic expression. And that's been shown to potentially be harmful here. So we don't want to do that. We want to avoid that. There are mutations here that are CBE targetable and I'll show you in just a second. And then again, we have patient cell lines available from our clinic to work on. And finally, this is a devastating disease. It hits usually as an early onset dementia in the 40s to 50s, and patients unfortunately usually pass away in 5 to 8 years very quickly. Okay, so choosing a mutation. How do we pick which mutation to target? So again, this is partly why we chose this disease. So here on the top, there are 234 described mutations here. About 95 of them are not base editor targetable, but 139 of them are base editor targetable. And of the ones that are targetable by base editing, it's about half and half CBE versus ABE. For the different mutations in CSF1R, there is one here that far exceeds the rest of them in frequency in the patient population. We don't know why that is, but that does increase the number of patients we could potentially treat for this particular with this particular therapy. So about 15% of all the patients have this mutation. So we designed guide RNAs to target this particular mutation. So these are the guide RNAs that were designed. As you can see, the PAM, the 3 base pair PAM, is at the end here, and there are different PAMs, but it also includes a wild type PAM, NGG, okay? So in our optimization experiments in HEC293T reporter cell lines, on the x-axis, you have different CBEs that we tested out. On the y-axis, you have on-target editing frequency. And the numbers here are pretty small, but the y-axis here goes up to single digits, double digits to 40, double digits to 50. Okay, so you can see that there is a large array of different editing frequencies based on which CBE you use. Here, we're using C4, C5, and C7. And if you go back to the previous slide, you can see that C4, C5, and C7 are not the wild type PAMs. So the data for C6, which is using the wild type PAM, the editing frequency is significantly higher, okay? And this is something that we do expect to see. Okay, so you pick the best CBE from these HEC cell studies, and you test them out in patient-derived primary cells. So the first cell type we tested them out in are fibroblasts, patient-derived fibroblasts. So it was pretty exciting to see that one of the cytosine base editors actually had an on-target edit rate of almost 90%, which if you do the math, this is basically equivalent to full engraftment in an allogeneic transplant. Next, we went into lymphoblasts because they are much closer to HSCs than fibroblasts. As you can see here, we had some editing efficiency problems with lymphoblasts because turns out they do not like having holes shot into them, and they do not like having DNA put into them. So here, the maximum editing efficiency we got in lymphoblasts, these are, again, patient-derived lymphoblasts, is about 20%, 16 to 20%. So not great. So I did an optimization experiment looking at mRNA delivery, encoding GFP. So you can modify the mRNA with different modifications to make it more stable. We also have a circular RNA here. And you can see that expression is much better in the lymphoblasts if you deliver RNA. So where we are right now, or what experiments are ongoing right now, is that we've made mRNA from our base editor and are transfecting the lymphoblasts with the mRNA. And again, after we do that, we are also optimizing the transfection in patient CD34 positive hematopoietic stem cells. Okay, so going back to the platform, animal studies, we're going to look at MIST-RG6 mice. So this is a xenotransplantation humanized mouse that basically allows human cell engraftment and human cell reconstitution. And the reason this mouse is so powerful, it has 7 different humanized genes, is that 1, you don't need a whole lot of human HSCs to engraft this mouse. And 2, it actually engrafts myeloid cells, or I'm sorry, reconstitutes myeloid cells at a near physiologic level, which is a struggle in the hematopoietic field. So this is a good model specifically for this disease because it's macrophage and microglia. Okay. Am I doing final? Okay. Okay. Let's look at functional studies. I'm just going to go over this really quickly to show you that there is a very simple functional assay you can do with this, and that's CSF1R phosphorylation. So you want something with an easy function assay that you can look at to say, yes, this protein is corrected, or no, this protein is not corrected. Okay, so main takeaways is that this is basically a platform that we are developing to hopefully treat microgliopathies by using the hematopoietic system, and the different steps are outlined here. But the hope is that we create a library of base editors and guide RNAs to be able to target various mutations for various microgliopathies. Okay, questions?
We have time for a few questions. Kevin, go ahead.
That's really interesting. Sorry, I was looking for the room for a while. What is the idea why this microglial mutation causes a leukodystrophy?
Yeah.
I'm just interested because, it just so happens that when we treat ALD, by putting the gene, correct the gene back in the microglia, we always thought it was a happy coincidence that it worked, but maybe there's more to it than that.
Absolutely. So the short answer is we don't know. We're trying to figure that out. In terms of how it affects microglia, we think, and I frankly don't think this is true for every single mutation that exists in CSF1R that's been described as pathogenic, but we think it changes the ability of CSF1R to autophosphorylate because it's a tyrosine kinase. And as a result, you have a problem then with microglial maintenance. So patients, if you look at their neuropath, have fewer microglia than the normal population. How that contributes to a leukodystrophy, I am not sure. But it could be that the microglia are responsible for maintaining myelin function and myelin health. And if you don't have microglia, then that homeostasis gets thrown off. That would be my hypothesis, but that's a total hypothesis.
And just a silly question, a simple question. And the idea here would be that if you have an HSC that's been edited, enough of it's going to get back and get into the CSF to replace or overcome. What is it going to do in the CNS?
Yeah. So the idea is that you do a regular transplant with the edited HSC, so it goes into the bone marrow, and then the bone marrow then produces monocytes. And because in the disease, the blood-brain barrier is disrupted, those monocytes will then traffic to the CNS across the blood-brain barrier and turn into macrophage-derived microglia-like cells and correct the disease that way. So, for example, in metachromatic leukodystrophy, that's now been shown to happen for kids who are transplanted with normal bone marrow.
One more question, and then we'll.
I'm wondering, could you talk about the editing? Are there any what approaches do you intend to take to minimize those?
Yeah, absolutely. To minimize those, it's more to see which editing strategy has less off-target. So it's going to be a massive screen. So the idea, because the off-targeting assays change so quickly and they're developed so with such frequency now, the idea is that we first look at the on-target and the bystander. We pick the best from those, best combinations from those, and then look at off-targeting together. for those best combinations.
Great.
All right, great, thank you.
While we're switching over, I'm going to introduce Peter Hadar, who's an epileptologist here at MGB. He's interested in the intersection between neurostimulation and neuroimaging. He studied at Harvard for undergrad, went to UPenn for medical school and residency, which we don't hold against him, because he then moved up here for his epilepsy fellowship. And he works in my lab along with other people looking at responsive neurostimulation and how that works, neuroimaging, applications of AI and machine learning, as well as stimulation approaches to understanding seizures and being able to control them better. He is moving on from the UE5 funding to funding through an AAN AES Susan Spencer Award for the next couple of years as he works to build a virtual neurostimulation model to enable us to predict better who should be treated and how they should be treated. Peter?
Thank you, Sid. And sorry for my voice. I was getting a little cold. So I'm going to talk to you today about mapping the structural and functional networks of temporal lobe epilepsy.
You got it.
Yeah. Just to hit on some main points that I want you to get from this talk. At the end of it, I want you to understand how a novel diffusion imaging technique and modeling can be used to model the microstructural changes in epilepsy. I'm looking to uncover the relationship between structural and functional connectivity during seizures. And I want to also discover how responses to neurostimulation, particularly through acute cortical stimulation mapping, can be informed by these microstructural changes in epilepsy. So the question is, why do we do this? And the reason I'm interested in this field particularly is because we started to use therapeutic neurostimulation in epilepsy in the last few years. It's relatively new and evolving, but the key thing is that we still don't 100% understand how it works, whether it's kind of a more acute thing or a more chronic thing. And there are a lot of theories, hypotheses, and it's an active area of research. Although in the past, epilepsy was thought to be more of a lesional-based problem where you have hippocampal sclerosis or you have some lesion, you try to find the lesion, remove it, patient gets better. It turns out that that is not entirely the entire story. And it's thought that epilepsy is more due to network aberration now that could be amenable to remodeling through things such as neurostimulation. However, the problem is that much of the neurostimulation approaches today still rely on this outdated lesional point of view for the most part, where you implant an electrode directly in where you think the lesion is and stimulate to try to get an effect. So the thought and we had was that if we understand epilepsy better from a microstructural level, then we develop more network-based neurostimulation therapies over time. And I hypothesized that the microstructural aberrations, these white matter connections between the gray matter that can be more finely resolved, would be very important to understanding seizure onset and spread. So during my UE5 and R25, which I started at Penn under Dr. Kate Davis and now continued with Dr. Sid Cash, I used an imaging technique called neurite orientation, dispersion, and density imaging, NODI, and investigated the role of the microstructural network in intracranial EEG and seizure onset and spread, which is kind of abnormal native activity to the brain, electrical activity. And my second one was looking at neurostimulation responses, which in a sense is abnormal induced electrical activity in the brain. And the main takeaway point, not to bury the lead, is that I did find, or I believe I found some evidence that microstructural abnormalities affect the propagation of electrical activity in the brain, much like macrostructural changes do, but are no means the entire story. So to talk a bit about the structural network specifically in epilepsy. So neurite orientation, dispersion, and density imaging, NODI, they have interesting acronyms, is an advanced multi-shell diffusion imaging technique. I used 64 directions and 3 shells that models brain tissue as 3 different compartments. So an intraneuronal, an extraneuronal, and a CSF compartment. I looked primarily at the FICVF, the intracellular volume fraction, which is kind of measures neurite density. We could also look at orientation dispersion index, ODI, which looks at how the fibers are arrayed in the brain. And you could also look at the basically how much free water there is in the brain, which is modified in diseases that involve atrophy and gliosis. NODI in epilepsy, so there has been research done in the past on just the imaging technique in epilepsy, and they've shown to I think no one's surprised, is that there is a loss of dendritic complexity and neuronal loss that you see in Noddy compared to the contralateral hippocampus in the case of hippocampal epilepsy, and when compared to healthy controls. It's a little tough to see, I think, in the picture, but trust me when I say that on the top side, on the right of the patient, there is some more darkness in the temporal pole, and on the bottom slides, you see some more darkness near the hippocampus on the left, and those signify decreased neurite density and increased dispersion. And one of the interesting things is that Noddy can be correlated to PET even when the MRI is negative, so it could potentially open up non-lesional epilepsy cases. And there's evidence as well that I will present in a little bit that these changes are maybe broader and widespread. And this is in comparison to DTI, which is you look at mean diffusivity and fractional anisotropy, which are kind of similar metrics, but don't get at the fine-grained resolution of the microstructure. So some of the work in the patients I worked on, I'm a co-author on this paper that came out last month in Epilepsy, where we looked at Noddy specifically in temporal lobe cases, and we looked at neurite density on the left, fiber orientation, and Madelin-Novis distance, which is kind of a multivariate Z-score. And what we see is that, just to highlight, in the hippocampus, the right hippocampus, more so than the left, but across all, just has decreased neurite density. There is actually decreased neurite density throughout the entire brain. The fiber orientation is less consistent, but overall, you do see significant changes in temporal lobe epilepsy patients, especially when you compare to controls. So bringing it together to structural and functional networks. So I was interested specifically in this idea of structural and functional coupling in epilepsy. It's a bit, use an analogy, if you have highways, which are the structure, and traffic, which is kind of the functional aspect, healthy people have normal highways without potholes and have a normal amount of traffic flowing through it. People with epilepsy have a lot of potholes in their highways, so stuff doesn't flow through as it should. However, during seizures, it's as if the carpool lane opened up, and now traffic can flow much more freely through a certain location, so it opens up. And the idea is that this is kind of how epilepsy works in a sense, where at baseline, patients with epilepsy have weaker coupling before and between seizures, but then during the seizure itself, the connectivity, the functional connectivity, is stronger, and with the coupling, and tends to go down the pathways that are more associated, that are kind of more normal and healthy. So I looked specifically at 40 patients who underwent intracranial EEG. I looked through 332 seizures, and these patients had DTI and Noddy, and I looked through them, and I used this method, this processing pathway, for folks who have done this before. It's kind of fairly standard, where I looked at the functional network during a pre-ictal, ictal, and post-ictal clip. I registered it to the brain in a brain atlas, so I could tell where the electrodes were in space and where the locations of seizure and spread were in space. And then I mapped functional connectivity over time. So I looked at connectivity maps at each 1 second windows, 1 second intervals, and marched them out from the pre-ictal phase to post-ictal phase to see how it would change over time. For the structural network, I looked at, I converted diffusion imaging and modeling. I registered it, and then I looked at diffusion tensor imaging and Noddy specifically. I created structural maps from those. Then I basically compared at every single second the structural map to the functional map and marched it out over time. And in that sense, you can actually build a change over time in the connectivity between the structure and the function as it marches through from the pre-ictal to the post-ictal state. And what I saw, at least at baseline, and this has been reported in the literature in the past, I think, with fewer patients, 7 patients and I think 40 seizures, but now it's 40 patients and 300 seizures, is that you do actually see a similar sort of change, that as you go from the pre-ictal state, when you look at diffusion imaging, which is kind of, especially the streamlines, which are kind of a metric for quantity, initially you start off as kind of a pretty weak coupling, then during seizure, the coupling increases, and then as the seizure ends, the coupling kind of decreases back to baseline. And you see this in the broadband networks, and you also see this a little bit in the alpha theta, which is 4 to 12 networks. You also see this mapped out. I have extra slides throughout the rest of the frequency bands as well. Noddy specifically, so looking at the neurite density, shows something incredibly similar as well, almost identical in some ways, in terms of the temporal change. that you do see initially that there is weak coupling, there is an increase, and then a decrease over time in the postictal state. And then you see on the far right is all the different sorts of metrics that you can derive from DTI and NADI. And you see that there is kind of a translation of overall the strength of coupling and the streamlines. But overall, the trend is still, the temporal trend is basically the same. And to try to hammer this point home and to kind of describe it in another way, if you look at the pre-ictal, ictal, and post-ictal phases, you do see increased structure function coupling. It increases during seizure, but in the magenta, I guess, is the NADI metric. And you see that it starts at a lower baseline and then it increases over the seizure phases. Just for kind of outstanding work on this sort of project, as I'm going in the next few weeks to publication, I'm looking at some of the other, I'm processing a couple more patients, but I'm also looking at localization, lateralization, and lifetime seizure duration in this regard. And you do see very similar changes across phase for all of these. I'm also going to, this didn't make it into the talk, but look at seizure spread specifically through electrode and region hopping of seizures. So now to interrogate the networks through kind of induced abnormal electrical activity and neurostimulation. So I looked at patients who underwent cortical stimulation mapping. It's usually used for diagnostic purposes to help delineate functional zones, especially in patients going to surgery, so you know which locations not to resect. However, when you do that, like throwing a pebble into a pond and you see a ripple, you see evidence of electrical activity spreading throughout the brain. And this can be termed as cortical evoked potentials. And the strength of these different ripples, I guess, throughout the brain can tell you a bit more about brain connectivity and its spread. So I looked, and this is a much smaller data set. I'm trying to increase the size, but I looked at 7 patients who had all the metrics of DTI, NADI, and CSEPs. And in this specific case, we did a train of over 30 seconds. We did 1 Hz stimulation at 3 milliamps. You see here on the right side of the screen, you see that we went through 30 trials and averaged them throughout. And the reason this was interesting is because there is a question, as I'm sure might come up, is how much is the microstructure aspect just an aspect of distance between parts of the brain versus just kind of connectivity? And when you see in CSEPs, for instance, when you give stimulation and you look at the response, it does appear very closely aligned. So the higher peaks in CSEPs tend to be closer to the actual spot. And in these graphs here, what I want to show is that kind of on the left side, I'm looking at the distance relationship between them. And over time, if you look at the Spearman correlation, you would expect as the seizures, as electrical activity spreads throughout the brain, that it's going to decrease over space. But you would expect it, so in DTI, it actually decreases exponentially. But in NADI, on the other hand, it's actually separate here in the orange. It doesn't decrease exponentially. So it seems to indicate that it's something more than distance, and it might actually be speaking to something biologic going on. I also try to look at local and distant connectivity measures. And overall, there is a lot more effects locally in these metrics, but you do see some slight relationship with distant connectivity measures as well. But this is, it's a very small population cohort. And then the last thing, I was trying to trace out CSEPs to the different metrics, and I was trying to look at the extent of connectivity between CSEPs and the different structures in the brain. And if you assume 1 is basically an equivalent amount of information of the CSEP network and the structural network, you see that for many of these, so fractional anisotropy, FICVF, which is neurite density, neurite dispersion is FISO, mean diffusivity, and orientation dispersion index. So if you look at all of these, you see that overall CSEP in general provides more information as to the overall spread of the CSEPs itself. Although for some of these, like FISO and mean diffusivity, they do appear to provide some more evidence about the network connectivity. And this is an evidence, the figure on the right as well shows NADI compared to DTI when combined. And you do see that even though this is a very small population, you do see that there is subtle changes, although not significant. And this kind of outstanding work, and why I think I want to increase the N just to analyze it further, is that I do want to eventually link up these acute DTI to CSEP measures to more chronic analyses. We started looking at some of our RNS patients. We currently have 160 RNS patients who are implanted with a wide variety of different epilepsies. And that's going to be the main focus, actually, of one of my next grant, the Susan Spencer grant on building a virtual neurostimulation model. And long term, I want to also think about pathophysiologically how neurostimulation works in epilepsy and beyond, and try to understand why electrical stimulation differentially induces seizures, stops seizures, and inhibits seizures, with the idea that by understanding this, we might be able to better understand and treat epilepsy. So with that, I'd just like to say thank you to the entire lab, Sid's lab, and also Kate's lab, Penn, for all their help throughout these years. Thank you very much.
Time for one or two questions, if you have Devin.
Yeah, that's great. The functional connectivity work, going to linear vehicle, getting on the carpool lane. One problem is that all the neurons are doing the same thing. Right?
Yeah.
So when you do functional measures, and they all look like they're highly connected with each other, it's hard to know, is that a real monosynaptic pathway, or is that just some polysynaptic pathway? Did you, I mean, I know this was depth electro, but did you, for instance, take epilepsy patients, look at connectivity of the surface imaging, compare it maybe to somebody who's having an electroshock therapy, whatever? I mean, that hypothesis would be maybe that they would find something besides connectivity inside of yourself.
Yeah, it's possible. It's tough because, as you know, there's no good way to get a set of healthy controls. I think other in different sorts of surgeries, even though like tumor or other things where they open them up, you can get kind of acute measures of connectivity. That would be another option as well. But yeah, I guess the problem with trying to use scalp is that the correlation between scalp and intracranial imaging has been pretty difficult to suss out. So all of this comes with kind of a grain of salt. And as I think I've shown as well, is that the structural and functional connectivity, although there is a link, it's not 100%, and it's not fully descriptive. So there's a lot of things going on there. Yeah.
I have a technological question. I'm sure I missed this when you set up. The body measure is a structural MRI method, right? How did you claim that before, during, and after the seizure?
So this is the so I actually just obtained it once, and it was before the seizure. And what we were doing is that's a stationary metric, and then you compare as the seizure evolves over time between that, and you see basically how that structural metric starts lining up with the seizure as it progresses. So you're comparing like a single structural metric and then just seeing over time as they kind of merge into kind of a similar connectivity measure. Yeah.
Thanks, Peter.
Sure.
Last but certainly not least is Simon. And we might go a tiny bit over here. Please stick around so that you could grill Simon when he's done. Simon obtained his MD and PhD from Heidelberg University in Germany as PhD researchers on mechanisms underlying central pain in animal models of MS. After completing a postdoctoral, after that, he went and did a postdoctoral fellowship with Mario Suva here in the Department of Pathology in the Center for Cancer Research, and has been working with him since as a neurology resident on his UE5, formerly R25, research grant. He's focused on deploying single-cell RNA sequencing technologies in human glioma samples and dissecting the molecular determinants of responses and resistance in highly treatment-resistant brain tumors. His goal is to leverage this information ultimately to discover new tumor vulnerabilities and dependencies, ultimately enabling the development of novel and rational therapeutic modalities for those biomarkers. Today, he'll be talking about responsive IDH-mutant gliomas to mutant IDH inhibitors and dissected by multi-omics and single-cell profiling.
Great. So the project I'm going to talk about, it was a collaborative effort between Mario Suva's lab here at MGH and Itai Taro's lab at the Weizmann Institute. I led the experimental part and a postdoc in Itai's lab. Avishai led the computational part. So the focus of this project was to look at IDH-mutant tumors, and with diffuse gliomas, who are the most common malignant primary brain tumors, we really differentiate two big groups. So one are the IDH wild-type tumors, which are sort of the typical glioblastoma, usually occurring in older patients and very aggressive. And then the other big group are the IDH-mutant tumors. The IDH-mutant tumors, they usually originate in younger people, usually in their 30s or 40s, and they start out as more low-grade and less aggressive. But with time, they become more and more aggressive and in the end behave similar to glioblastoma, and unfortunately also are always fatal with very limited therapy options. So the IDH-mutant tumors have actually a very interesting biology. The class-defining mutation is the IDH mutation, and it's shared by all cells within a tumor, so it's clonal. And most likely, it's the tumor-initiating and first mutation in the tumor. IDH sort of was a very unlikely candidate as a tumor-initiating mutation because it's not a typical oncogene and also not a typical tumor suppressor. So you might remember from biochemistry, IDH is an enzyme in the Krebs cycle, and it takes isocitrate and transforms it into alpha-ketoglutarate. The mutant IDH actually does not lose its function, but it gains a completely new enzymatic function. And basically what it does now is the mutated form takes alpha-ketoglutarate and makes a completely new molecule, 2-hydroxyglutarate, that does not exist in the healthy human body at any concentration. And it produces really high concentrations of this 2-hydroxyglutarate. And what happens is that 2-hydroxyglutarate, because it's so similar to alpha-ketoglutarate, it will inhibit enzymes that use alpha-ketoglutarate. And many of those enzymes happen to be DNA demethylases. So it leads to a DNA hypermethylation. So it sort of changes the epigenetic state of the cell, which ultimately leads to a malignant transformation and then to tumor formation. So the discovery of the IDH mutation was really exciting because it's a very attractive therapeutic target. First of all, it's present in every cell of the tumor because of clonal mutation, but also it's a highly druggable target. And the field has developed many different small molecules inhibitors of IDH. In the beginning, the clinical data was sort of disappointing because we first tried it in the high-grade, late-stage form of IDH mutant tumors. And really the breakthrough was in a recent phase 3 trial where we tried it specifically in low-grade tumors. Most likely it's effective in low-grade tumors because in the beginning the tumor is still dependent on the IDH mutation, but once it accumulates more driver mutations, likely the IDH mutation becomes a passenger and it's independent of IDH. So in this trial, actually, there was very little objective radiographic response. So only about 10% of treated patients had tumor shrinkage on MRI, but there was a significant improvement in progression-free survival as defined as stable disease on MRI. And that sort of also matches our clinical experience. So as an example, to take one of the tumors that we actually profiled in our study, BWH445, it's sort of a typical case for oligodendroglioma, so a subform of IDH mutant tumors. So this patient was diagnosed in 2003 and got the standard of care, which is basically tumor resection followed by alkylating chemotherapy. He then was tumor-free until 2012, where he suffered recurrence, and that's typical. So those tumors always eventually recur. And after recurrence, your options are really somewhat limited, but they chose to re-challenge him with temozolomide, so alkylating chemotherapy. And he was then tumor-free until 2018 and had then an earlier recurrence. And it's also pretty typical that as those tumors recur, the recurrences come faster and faster. And eventually the tumor becomes very aggressive and hard to treat. So in 2018, he was enrolled in a clinical trial of IDH inhibitor and went on IDH inhibitor treatment. And we have been following the patient since, and he didn't see any tumor shrinkage, but he has been stable on treatment for now almost 7 years. And that's quite exceptional. Usually at this stage, you would expect recurrence within 2 to 3 years. So that's really reassuring. And as you can see, after treatment is not really an objective response per Wano, but you do see stability for many, many years, which is really exciting because it shows for the first time that we have in primary brain tumors a targeted treatment in diffuse gliomas. And also it raises the possibility of durable responses. But it also raised a lot of open questions. So the mechanism of response to IDH inhibitor was really not clear. Because it doesn't seem to make the tumor shrink, but more stabilizes it. And then we also don't understand why some people have excellent responses, while other patients don't respond at all. And then also radiographic endpoints might not be best to evaluate treatment response because you rarely see a tumor shrinkage, even though the patient might stay stable on IDH inhibitor for many years. So we wanted to address some of those questions, and our approach is really to try to evaluate the response directly in patients on inhibitor, which is quite difficult because, as you can imagine, like in our patient WH445, once he went on the inhibitor and then was stable for 7 years, there's very little indication to re-resect the tumor on treatment. But we were fortunate that there was a phase 1 trial where per study protocol, patients got 4 weeks of IDH inhibitor treatment and then underwent resection. So we could get tumor tissue while the patient was getting the IDH inhibitor. And we were able to get patient tissue on inhibitor for 3 patients in this trial. For 2 patients, we had post-treatment tissue, and then for 1 patient, actually our WH445 patient, we had both tissue before starting the inhibitor and then after starting the treatment, so that we had a paired sample and could compare directly in this patient. The way how we analyze those patient tissues is by single-cell sequencing. So basically our workflow is that we collect the tumor tissue directly from the resection, then split it into single cells, and then perform single-cell sequencing. That's quite powerful because you get the transcriptome of each individual single cell, so you can evaluate the intratumoral heterogeneity. And Mario Suva's lab has done this previously in IDH-mutant tumors. And they were able to show that IDH-mutant tumors do really have this very satisfying developmental hierarchy and follow the cancer stem cell model quite closely. So you have basically a small population of stem-like cells who are really the proliferating cells in the tumor, who then differentiate towards 2 axes of differentiation, one that's more astrocytic-like and one that's more oligodendrocytic-like. That's not true differentiation. So those cells, they stay tumor cells. It's sort of a twisted version of normal development. So we analyzed those 3 samples, and for the 2 patients where we only had post-treatment tissue, we combined those with 6 untreated samples just so to have a comparison with untreated tissue. So now we get the transcriptome of each individual cell, and the first step is to identify the malignant cells, because one of the main advantages of single-cell sequencing is that you can analyze the malignant and non-malignant cells individually. In this case, we defined or identified the malignant cells both based on clustering. So in the single-cell data, cells will cluster together based on the similarity of the transcriptome, and then also expression of canonical marker genes and typical copy number variations. All 3 of our patients were oligodendroglioma, which is fortunate because oligodendroglioma has a canonical disease-defining copy number variation, 1p19q codeletion, and we could show this in all our cells that we identified as malignant. So we have high confidence that we actually separated malignant from non-malignant cells. So the next thing we did is we compared differential gene expression between our treated and untreated malignant cells. And the main thing that stood out is that after treatment, you have an upregulation of gene sets of normal astrocytic development and a downregulation of the more stem-like gene sets, or gene sets for oligodendrocytic differentiation. So that was already very interesting, but it doesn't tell you yet if that's just a general upregulation or a general transcriptional change across all cells, or if there's really a shift in the cell fraction. But this is actually something that we can answer very well with single-cell sequencing, since you have a transcriptome for each individual cell. And what we found is, if you remember this developmental hierarchy with the stem-like cells that differentiate toward AC and OC-like, we found that IDH inhibitor treatment really depletes the stem-like cells, but also the OC-like cells, and increases the fraction of the AC-like cells. And as you might imagine, as you deplete the stem-like cells, also the number of cycling cells, or a fraction of cycling cells, is decreasing. So this, and that was both true for our unmatched sample and then also for the matched pair. So that was quite interesting to us, because it raises the possibility that maybe the inhibitor treatment is actually pushing the more stem-like proliferating cells that really drive the tumor to differentiate more towards the astrocytic lineage. And we also confirmed those findings by ISH in situ hybridization, just to have some autorenal evidence. And it basically showed the same thing, also an increase in the astrocytic differentiation and decrease in the stem-like and cycling cells. So that was really exciting, but one of the main limitations of our study was the small sample size. So we only had 3 treated samples, and it's really hard to recruit more samples on treatment. So what we did is we tried to validate our findings in larger bulk cohorts. So this is the study from which we got our 3 on-treatment samples, and it includes 21 treated patients for which we had bulk data. So we could go back and then interrogate the bulk data with transcriptional profiles that we identified with a single-cell study to see if it holds up in this bigger data set. And when we interrogated the bigger data set of 21 treated patients, we saw a similar effect. So IDH inhibitor treatment seems to increase the AC-like cells and decreases both the stem-like cells and cell cycle. And also in this data set, AC-like differentiation and cell cycle, they're anti-correlated, which is kind of what we would expect. Next, we also wanted to compare our data set to a larger sample of untreated IDH-mutant tumors. So we went back to the TCGA, so a large data set of bulk data for 134 untreated IDH-mutant tumors. And basically what we were showing here is that our 2 treated samples are very strong outliers in terms of the strength of their AC differentiation compared to this large cohort of untreated samples. So MGH170, one of the samples, was by far the most AC differentiated, and then also MGH229 was top 3%. Lastly, we also used a novel mouse model to confirm our findings. That's a mouse model that Jerome 14, one of our collaborators, has developed that's still unpublished. It basically based on introducing both the IDH mutation and then also loss of p53 into the oligodendrocytic lineage in mice, and those mice will develop IDH mutant tumors. And we first basically confirmed our findings that with IDH inhibitor treatment, also in this mouse model, we see an increase in the AC differentiation and then a decrease in the stem-like and in the cycling cells. But the more exciting thing is that in this mouse model, we are also able to leverage multi-omics. So basically, we can do single-cell gene expression and also methylation sequencing. And the first thing we could show here is that with IDH inhibitor treatment, you have a moderate but significant decrease in the global methylation of your sample, which is sort of what we would expect. So if you inhibit 2-hydroxyglutarate, the DNA demethylases should do the job again, and you would expect that the hypermethylation we usually see in IDH-mutant tumors is at least partially reversed. Next, we could also see which genes are differentially most demethylated and most increased in expression in our treated samples. And we found that the gene list that's most demethylated and highly expressed in our samples is strongly enriched with genes for astrocytic differentiation, which fits very well our hypothesis. So together, all of this made us converge on a model where we think that IDH inhibitor is not a cytotoxic treatment, but actually acts as a differentiation treatment. So basically, it forces the stem-like cells by sort of normalizing the epigenetic status to differentiate towards the astrocytic lineage and decrease cycling in those cells. That also explains why we rarely see tumor shrinkage, but often find that patients stay stable just because the tumor is behaving more like normal brain tissue. So this was really exciting, but there's still a lot of unanswered questions that we try to answer. So first of all, why is it, or what are the mutation that make people or tumors resistant to IDH inhibitor? And there are a few candidates that we want to pursue. Then also, what's the best way how to integrate IDH inhibitor into standard therapy? Because IDH mutant tumors, different from glioblastoma, they're actually fairly chemosensitive. So how do you want to sequence it? Do you want to use the IDH inhibitor in the recurrent situation, or should you use it upfront? And then also, can you ever stop treating patients with IDH inhibitors? So in BWH445, you can imagine he's now stable since 7 years. Do you have to continue the IDH inhibitor indefinitely? And then finally, are there any better biomarkers than imaging to assess if patients are responding to IDH inhibitor? Can we somehow maybe leverage astrocytic differentiation to show if a tumor is responding to the IDH inhibitor? Overall, it's a very exciting development and very exciting time to be in neuro-oncology, but still a lot of questions to be unanswered.
Tumor biology escapes me, and I'm still want to hear a little more about your theory as to why it works. So if I'm saying that it's a very early stage mutation that's sort of allowing the tumor to happen. Tumor's already happened, so I would have guessed you can do anything you want to the IDH, and it wouldn't matter at that point.
Right.
That's whatever, the creature is out of the barn. But that's not the case. So are you reverting it? Is basically the tumor reverting in a way that you're getting cell replacement because the cells that are replacing are much more normal astrocytes? Is that what's going on?
So that's a really good question, and the way how we think about is that the IDH mutation probably happens in a progenitor more stem-like cell and leads to a differentiation block, which keeps the cell locked in a more highly proliferative, more malignant-like state. And most likely you need a few more mutations, probably at least P53, to transform the cell. But the cell still has not accumulated a lot of additional driver mutations. So it's still sort of addicted to or dependent on IDH to stay in this more stem-like state. And that sort of explains why those tumors usually start out very low grade, very slow growing. And then as they accumulate more mutations, they become more and more aggressive. So probably there's a time window where you're still dependent on IDH. But as you say, as you develop more mutations, likely you don't need the IDH mutation anymore. And that's actually often what we see, that the IDH mutation in 2-hydroxybutyrate, as the tumor progresses, becomes more of a liability, because 2-HG is sort of slightly cytotoxic. And often as the tumor gets more aggressive, it will sometimes just kick out the IDH mutation. But probably there's a window early on when the tumor is still low grade, when you can treat it. But it's very interesting to ask what are the mutations that actually make the tumor resistant or independent of IDH, and it's sort of the next steps.
So you said this was a germline mutation in IDH, or is it a somatic mutation?
It's a somatic mutation.
So can you detect the unique metabolite in the urine or blood or anything?
So you cannot. There's actually a germline version that's quite rare. Those people also develop brain tumors, and in that case, you can detect it in the urine. And with the somatic mutation, you develop very high concentration in the brain, but not high enough to detect it in the urine. But actually, since the concentration in the brain is so high, you can actually use MRI to, on imaging, see the 2Hg. So there are certain new MRI sequences that are being developed to diagnose it based on imaging, but we don't yet have a good blood or urine biomarker. It's not in high enough concentrations outside of the brain.
Most of the mechanistic ideas for IDH mutations are running to, you seem to support using the inhibitors as early as possible. I know, other than just having done the trial yet, what are the arguments that you want to wait?
That's a great question. So at the moment, we really don't have good data what's the best sequence. And the argument to do it upfront, as you say, is because you think that early on it's more effective, and you risk if you wait that you accumulate more mutations might not be as effective anymore. The counter argument is that the big phase 3 trial was not done against the standard of care. It was done against just observational treatment. And those tumors, they are actually very chemosensitive. So as you saw BWH445 was stable for 9 years after standard of care. So the question is, if you give the IDH inhibitor upfront, will they still respond as well to standard of care and once they progress after IDH inhibitor? At the moment, sort of, we try to stratify patients that where the tumor can be completely resected, looks low grade, so a low risk patient. And there we want to delay radiochemotherapy to reduce toxicity. Those are sort of the patients where we use it at the moment, sort of to delay toxicity from standard of care. But it's sort of just assuming that the standard of care will still work later on. And that's a big if. So that would be, say, well, we have a well-defined standard of care. We know it works. So let's keep the experimental treatment for when the tumor actually becomes resistant to the standard treatment.
I think you said the IDH mutation impacts the I was just curious, does that also leads to eventually increasing gene expression? And then I was just wondering, does that also affect like chromosomal modeling and such?
Sorry?
Like the modeling.
Yeah, that's a great question. So that's really open. It definitely does. We don't really understand it very well yet, but there was one paper a few years ago from Brad Bernstein's group that did show that IDH mutant tumors, they have disturbed chromatin remodeling and enhance the dysfunction. So basically the separation between different enhancers breaks down. So there's definitely also some change in the three-dimensional structure of the chromatin. But we don't really know yet how the IDH inhibitor would affect that. But that's a super interesting question.
Well, thanks very much, and thanks to all three speakers today, and thanks for everybody for sticking around a little bit.
Thank you.